Welcome to another episode of Progress, Potential, and Possibilities, discussions with fascinating people designing a better tomorrow for all of us. I'm your host, Ira Pastor.
Welcome, everybody, again to another episode of our show, bringing another really fascinating guest today involved in creating a better tomorrow for all of us. As a little background to today's show, recently the Novo Nordisk Foundation, the Wellcome Trust, and the Bill and Melinda Gates Foundation announced the formation of something known as the Gram-Negative Antibiotic Discovery Innovator, which is an initiative that will focus on the discovery of these direct-acting small molecule antibiotics with broad-spectrum activity against the Enterobacteriaceae family of bacteria, which includes Salmonella, E. coli, Shigella, as well as Klebsiella, which has been selected as the initial target of the program. The program also aims to address the lack of antibiotics for various gram-negative bacteria, as well as the public health threat posed by AMR, which has been a very important topic for us lately. And as such, we have the honor today of being joined by Dr. Marianne Holm, who is Vice President and Head of the Infectious Disease Program at Novo Nordisk Foundation. There, she is responsible for supporting the development and implementation of various strategic initiatives and research programs, developing partnerships, ultimately that contribute to the impact and visibility of the Foundation's activity in infectious diseases, as well as supporting their various cross-disciplinary initiatives in AMR. Dr. Holm is a medical doctor. She's also an epidemiologist. She previously led the Department of Epidemiology and Public Health Research at the International Vaccine Institute in Seoul. They're responsible for coordinating and implementing some very large programs funded by the Fleming Fund, focused on AMR surveillance in low and middle income countries. Prior to that, worked several years at the School of Public Health, University of Hong Kong, and has been a member of various groups focused on this issue, including the WHO Technical Advisory Group and Coordination Group on Vaccines and AMR. She did both her medical degree and Ph.D. University of Copenhagen, master's in science focused on epidemiology from the London School of Hygiene and Tropical Medicine, University of London. And we're honored to have with us. A lot to get into today. Dr. Marianne Holm, thank you so much for taking the time to come on the show today.
Well, thank you for inviting me.
Yeah, wonderful to have you. Really looking forward to getting into this important topic. Before we get into infectious diseases, though, I would love to, as we typically do, sort of hand things over to you just to talk for a few minutes about your background, because I noticed in reading through a lot of your earlier work, you started off not in infectious diseases, but in cancer epidemiology, both breast cancer and a little bit in colon cancer as well. Your PhD was, looking at the Denmark lifestyle, sort of the Danish diet, cancer, and health cohort was related to various cancers. Tell us a little bit more about yourself, a little bit of your background, and how you got started in this particular area.
Yeah, well, thank you. Well, as you mentioned, I am educated as a medical doctor many years ago now at the University of Copenhagen. And actually already when I started my studies, I was very fascinated by sort of the more public health matter of things and sort of taking a more general approach to understanding medicine and how that affects not just individuals, but also populations. And so I already started having an interest in epidemiology, as you mentioned, but certainly also medical sociology and there through also sort of the methodologies and understanding statistics of large population sampling and how we can use research at a population level to understand much more about how will the overall effects of what we're trying to achieve in medicine actually have the most benefit. So So I started already doing research, and that was actually the breast cancer. I worked part-time as a surgical assistant in one of the large breast cancer units at a hospital
Yep.
here in Copenhagen, and through that got introduced to a number of quite passionate researchers and professors that were doing very interesting research. And so I started already my first research projects and writing my first articles in back when I was a medical student. And I actually didn't end up spending a lot of time in the clinical setting. I did a few of these sort of internships and things like that, but very clear to me from an early point on that I was very driven by research and doing that. So that's why I actually started pursuing first the degree in epidemiology. I wanted to see, you learn a certain amount of epidemiology in medical school, but then there's much more also methodologically, scientifically, there's much more to learn. And so I was interested and curious about that. So actually during my first maternity leave, I started signing up for some courses at the London School, and then quickly got really fascinated by that and ended up doing a whole degree there
Yep.
while I was taking actually a maternity break from the clinical setting. And in the end, then I actually started connecting back to some of the professors that I had worked with previously. By that time, I'd actually moved abroad with my family. So I was sitting in Asia already at the time, but connected back to some of the professors and started doing the epidemiological cancer research that you also described. And that's also the good thing about epidemiology and working on population health data. The type of research doesn't require you to sit and go to a lab every day, but it's something
Right.
that you can do on the distance a little bit more easily because you work with large data sets with doing statistical analyses and things like that. So that's how it all started and how I ended up becoming part of that research group that they had at the Cancer Society at the time. And again, it was kind of this thing. I didn't start off on a PhD. I started off just doing the research and learning new things and did a number of papers. And at one point, the supervisor I was working with there said to me, well, why don't you actually write up into a thesis and submit it? And you've written so many papers that's actually 1.5 PhDs already. So why don't you go ahead and and get your degree? And so it's for me, it's always been the curiosity and the sort of the passion for it much more than, is it going to lead to this or that? And so I ended up finishing the PhD from the University of Copenhagen as well. But actually, most of the time I was living and working in Asia, and actually at the time then also working at the School of Public Health in Hong Kong. So I sort of
Mhm.
continued down that path and actually never looked back. Sometimes you had this, should I go back into the clinical setting? Should I get a clinical specialty? Because at the end of the day, when you've gone through medical school, that is really, what you're trained to do and what you envision you're going to be doing. But at some point in time, I'd sort of, I had picked my path and found out that this was my thing.
Excellent, excellent. And from there, as I mentioned in your bio, you head to the International Vaccine Institute in Seoul, and about a year and a half or 2 years ago now, we hosted Dr. Jerome Kim on the show, and we talked about streptococcus and cholera, and we'll be talking about obviously antibiotics a little later on, but we forget sort of the vaccination side of this, and I found it very interesting that in your time at IVI, you spend a lot of time, about on typhoid and typhoid vaccine. This is one that we don't get here in the United States, but nonetheless, it's a traveling vaccine. And you spent, again, in this case, it was just quite interesting because you were studying not just surveillance, but sort of the introduction of the typhoid conjugate vaccines, looking at both Ghana and the Ghana healthcare system, as well as Madagascar. So quite a distance there, but different populations and insights. What did you learn at this point? Because this is a quite interesting area that, again, when we get to the AMR topic, right, there's a lot of different aspects of it that we're going to have to bring together to solve this really major problem. But again, we'll be talking about antibiotics, but some of your learnings from your typhoid days.
Yeah, so I switched back. So I worked a lot with sort of the more non-communicable disease type of epidemiology, both with my PhD, of course, but also when I was at the University of Hong Kong. But always was very, very intrigued by understanding, not just single associations and what does this risk factor lead to this disease or whatever, but just understanding everything in a context and understanding that there's there's not like one thing you can separate out, because people are complex organisms, societies are complex. So it's always been about, overall, how can we work better on things, and through that become a bit of a generalist, but also someone who's been quite curious, so quickly to move around. And so I actually did change the focus a bit from the non-communicable disease epidemiology that I was doing in Denmark and Hong Kong. And at IVI, now it's full on infectious disease epidemiology. But still, vaccines, hugely important prevention tool at the public health scale, because this is something we actually use to prevent infection. So really very nice to be able to then start working in that field. And IVI is such a unique organization, and very well placed to deal with a lot of the topics like typhoid, a lot of the diseases that are not of particular high commercial interest, but still hugely important for many, many populations. And so it was really, very nice opportunity to be able to go and join this organization that sort of really works to focus on these neglected areas, but still take a very scientific approach and try to develop really good products. And then at a certain point in time, be able to deliver something and tech transfer it to industry, which at that point in time, because somebody has made the upfront investments and proved that this is effective, as was the case with this typhoid vaccine, we were able to. This was something that was developed, all the way through at IVI. But then at a certain point in time, you're able to bring in industry partners, because now you've done sort of the de-risking and the proof of concept at an early stage. Then you can bring in industry partners, because you do need industry to then also eventually pick this up, manufacture, not just for clinical development, but of course also in the other end when a product is hopefully successful and can be marketed. So then it was really interesting to work with the basic science and to work on this whole process of trying to get industry interested in this and reach a certain point where then we can actually prove also through the clinical development that this is a product. And then the tech transfer was done with the condition that these industry partners would sign up to really make this vaccine accessible and affordable in those populations that need it the most. And that was kind of a very, as you can imagine, that's a very nice way to be able to work under that, so that you understand that the market is important to drive things as well, in order to be able to manufacture and produce and get things delivered. But at the same time, that you have an opportunity to actually make sure that the products that you do help develop can benefit those who need them the most. And so that was a truly inspiring organization to work for, because that is really the remit of what IVI does. And particularly with the typhoid, it was interesting because from an epidemiological point of view, it was also equally important to understand, so what is the burden of this disease? It was widely known for many years that it was a huge problem across South Asian countries, for instance, but we didn't really know at the time how much of a problem is this actually across the African continent. And this is not something that I personally worked on. This happened before I joined IVI, but there was a strong epidemiological team there that worked to really prove, well, this is actually a growing problem in the African continent as well. And interestingly, we could also see that there were a bit of a difference in which are the sort of dominating subtypes of the bacteria on the different continents. And so it's really important to see, so the vaccines that we are now developing, would they also be useful to be used in Africa? And we could see that they were. And then after the vaccine was introduced, we were able to do these huge studies to show that it actually did make a difference. But with epidemiological analyses, we could say, and in which parts of the country and which parts of the population would this have the biggest impact. And so then you can really also target it so that you can reach those in highest need.
And it is part of this journey at IVI that, along with your mapping and surveillance and the various tools that you're using for all the aspects of your work, that AMR, antimicrobial resistance, sort of pops up into your published literature. You begin exploring things like national reference laboratories, food safety laboratories, human and animal health facilities. Could you tell us just about sort of the initial moment where this came onto your radar as a problem per some of the surveillance work that you were doing in the field?
Well, I mean, AMR or antimicrobial resistance has been something I have known even since I was in medical school, because we already started experiencing that patients were getting increasingly difficult to treat because
Yep.
all of a sudden they would get infections with bacteria that were no longer susceptible to the sort of the regular drugs. And so we saw that already when I was in medical school, I remember, and also when I worked in the clinical setting shortly. So it's something that I've always in my adult life at least been aware of. But obviously it's something that has been growing in scale. And then when you move to Asia and live there and you work across a number of different countries that are struggling a lot with this on a day-to-day basis, it becomes very clear that it's a very complex problem. And, like you mentioned, food industry, you mentioned animal setting, it's something that really cuts across a number of sectors. And resistant is not just in one type of bug or disease, it's in multiple different ones. So it's a little bit harder to sort of address like some of those big unilateral programs like an HIV program or TB program, where you can sort of focus on one bug and one disease. Makes it, I'm not saying that simple, but that at least is a little bit easier for people to comprehend, and you can sort of focus that on a certain group of patients to a certain degree at least. But with AMR, it's so many things, and there are so many contributing factors, and there are so many things that are at odds with each other to really tackle this problem. So it's something that actually cuts across everything we do in infectious diseases, no matter what pathogen we focus on or what intervention we focus on. Somehow it all, in a one way or the other, will have this issue or relate to this issue in one way or the other. The work at IVI that I actually spent most of my time on when I was at IVI was building up through, as you mentioned, the Fleming Fund or UK aid-funded projects, was to really try and get a much better understanding globally of what is the scale of AMR, what is the problem, what are the dominant pathogens that we see, what should be the primary focus. And I think that's also an attempt to try and tackle this very complex problem, is that we need much better data. And so this is something that was really championed a lot by the UK government at the time, to sort of say, okay, we actually want to establish this fund that is solely related to getting much better data on antimicrobial resistance, because we have an idea that it's really bad, but we actually don't know how bad it is. We don't know where the problem is the biggest. We don't know what the biggest problem is within that. So it was really much this idea of getting much better evidence and much better data. So we were funded to support a number of countries across the Asian region, where we worked directly with the ministries of health to try and understand what kind of data had they already been collecting in labs. We actually went out and identified a lot of just logbooks with rows and rows of data, but when it's just sitting in those logbooks, you can't really use it for anything. So a lot of the projects were also trying to help countries digitalize the lab, the reference lab work. It was also, as you mentioned, capacity building to make sure that the data that they do collect was of the best possible standard, so they can really start using this data for improving their own surveillance of the problem and then informing what to do about it. So that was really sort of trying to build up that baseline of better laboratory capacity, but certainly also much better data handling and analysis capacity across that region. And that has then also led to much better data feeding into this global burden of disease program, and the publications from that. So we were not the only ones doing it, but a number of the Fleming Fund programs that were supported was also the global burden of disease study done by the IHME and the Graham study group in Oxford. So they were also going around and trying to sort of collect all the data that was there. And that's where we have those numbers from and how we contributed to those studies. But I think it's also important to mention that one of the main findings from those studies was that we still have too poor data from a lot of regions in the world that could use with getting more support in ramping up surveillance, getting much better data to inform how they work from the national level, but certainly also locally in hospitals. How can we get better data on how we should prescribe antibiotics, etc.
Excellent. Perfect. Appreciate that background. Okay, so now with all that, and again, we'll get to the gram-negative antibiotic discovery innovator in a minute, but would love to just again come back to Novo Nordisk Foundation now. And again, we a couple of years ago spent time with Dr. Thompson, talking about the foundation, and obviously we know Novo as a diabetes company and as obesity company and so forth. Not necessarily an infectious disease company, but it's a major focus of a major focus of it is reducing the burden and threat of these infectious diseases. And as Dr. Thompson told us then, look, we're a pretty big pharmaceutical company. Although we don't have that as a therapeutic focus, we have a lot of the great tools that can help. Since then, it seems like your portfolio of these programs is quite large, everything from pandemic antiviral discovery to interesting work on the agri infection side, coronavirus initiatives. Talk a little bit about the foundation, your role as head of infectious diseases, and a little bit of sort of the portfolio of these different initiatives that we have, and then we'll get to antibiotic discovery.
Right. So, the Novo Nordisk Foundation, I'm from Denmark myself, as you know, and so I was really very pleased to be able to come back and join this foundation at a very exciting time. So I joined about 3 years ago, but this is a very old foundation. It's more than 100 years old now. And it has obviously grown a lot in size recently. It may be important to make the distinction that the foundation sort of sits on top of what we call our holdings company, and holdings then is the majority shareholder in both the Novo Nordisk company and Novo Nordisk, which are the two sort of larger companies, and then about 170 other biotech and life science companies and a few other investments too. And so the way we operate, us who work in the philanthropic arm of the foundation, we're benefiting from the fact that these companies are doing great, or Novo Holdings is doing really well, and some of the companies, of course, also as is known. And we benefit from the fact that they share a certain amount of dividends into the foundation, and us who work on the philanthropic activities, we get to use that to support research and innovation, and also humanitarian and social purposes. And so we don't directly work with the company, but we are, of course, benefiting from the fact that they're doing really well, and part of the overall Novo family. But traditionally, as I said, the foundation is more than 100 years old. Traditionally, it's been a lot smaller in what we have been handing out, but it's also been a bit smaller in scope, focusing very much on the Danish and Nordic region, very much on research. The medical focus has been primarily on diabetes and also cardiometabolic disease. But in recent years, we have, with the new 2030 strategy, we've really grown in scope of what we focus on. And as you mentioned, we're moving in and have a strong pillar within sustainability, where we really focus on solutions for better food system or sustainable food systems, but also for the green transition of society. We have, as we've always had, but have grown within what we call the life science ecosystem pillar, where we support a lot of basic research and science, but also science and STEM education and innovation. And then finally, within the health pillar, we're now 4 program areas. We're still the traditional and sort of legacy program, which is the biomedicine with diabetes and with cardiometabolic disease focus. Then we have an obesity and nutrition focus that also supports research in this area. Then we have our social and humanitarian department. And then finally, we're the infectious disease program area as one of those 4 areas within the health pillar. And we're a relatively new program area at the foundation. So we were only established in 2022. That's not to say that the foundation hasn't been supporting research in infectious diseases for a number of years already, because that has happened through some of the traditional sort of, for instance, biomedical or clinical medicine programs. And also Novo Holdings has actually also been managing a few of the initiatives within AMR, but I can also come back to that later. But now this has been the first time where we've had a very designated focus area, which solely focuses on infectious disease, support research in this and support a number of initiatives. So that's what, I and the whole team that work in the infectious disease program, we've been part of building that up within the foundation. So it's been really exciting to be able to sort of broaden out the scope within our area and make sure that the foundation actually works very actively in this area as well. Yeah.
Perfect. And with that, it brings us to the gram-negative antibiotic discovery innovator, announced just a couple of weeks ago. So Novo Nordisk Foundation, Wellcome Trust, Gates Foundation. Take us a little into the story of setting this particular, because I know it's the challenge. We'll get to the details of the challenge, but the challenge, it goes to sort of the grand challenges site that the Gates organization runs. But talk a little bit about the background of it, setting it up, and then you talked before about looking at the big problems, and Klebsiella, although it's not top of mind to most of us, it's a big one. And again, I'll let you go into this. We've not been successful creating vaccines. It's gram-negative, obviously, with its own issues. all sorts of multi-drug efflux pump resistance capabilities, and it produces this nasty slime, quorum sensing stuff, which make it resistant to all sorts of sterilization techniques when we talk about outside the human situation. Talk about how, you can obviously, I understand why you chose it, but talk about how you chose it.
Yeah. So as mentioned in infectious disease program area, we sort of been building out a strategy, and it's no secret that we have sort of a big focus on AMR within that. And then we've also, as part of this broadening out the scope, we've also, as a foundation, we're working also very hard now to sort of broaden out our reach and start working more internationally. And that's something that's of particular relevance to our area, where a lot of the things that we try to solve is actually much more prevalent in other parts of the world, and there are endemic settings where the best research is done and things like that. So we are, by virtue, you could say, already a very internationally focused program area, but it's something that the foundation as such also really has been interested in doing. And as we do so, we really want to go out and make sure that we identify the areas where we can have the largest impact. We also want to work and learn from existing entities in the world that have a good experience with this. And that's one of the reasons why we've decided to partner up with exactly the Gates Foundation and Wellcome Trust, which both have huge legacies within infectious diseases and supporting research internationally in this space. So that's why that's obviously a natural for us to partner up with these entities. And very exciting for this tripartite collaboration that was established just last year, where we've defined again different pillars of work, but infectious diseases is one of them. And this is sort of the first initiative that has been announced under this tripartite collaboration. And that was really what we all felt. AMR is such a huge problem. AMR is something that we all need to use, not just pool our funds, but certainly pool our voices and focus. And so this has been designed as an effort together with the three foundations. And both, well, I guess all of us, but certainly our foundation, we are traditionally also very much focused on basic science, on early research that can really try and solve for some of the bigger problems. And so it was natural for us to start with a focus there. It's also building off of an experience that the Gates Foundation have with building these drug discovery accelerators, that they've done it also for other programs like TB and malaria. And the Wellcome Trust, I think actually out of all of the three foundations have been the most active within AMR and have worked across a number of areas, and also traditionally very much funding basic science. So it was a natural thing, I think, for us three to come together. And as you mentioned, the gram-negative bacteria within the huge problem that is AMR, the gram-negative bacteria definitely are responsible for the largest burden and also the hardest nut to crack, so to speak, the biggest scientific challenge. As you mentioned, the gram-negative, and if we just take Klebsiella as an example, these are particularly difficult bacteria to treat. They have double types of membranes that act in opposite ways, which means it's often very not just about identifying drug targets. It's also about figuring out how to actually get the drugs to be able to pass both types of membranes and also avoid being effluxed out, as you mentioned, because some of the resistance mechanism is just quickly sending out the drugs again. So you want to also make sure that the drug actually is able to get into the bacteria, to stay in there long enough to have its action happen. So there's a lot of mechanism just to actually make something effective against a gram-negative bacteria like Klebsiella is a huge challenge. And that's one that we really want to try and tackle because, I mean, as the recent GBD Lancet study stated, if we were actually, they make these different scenarios and basically said, if we are able to tackle this, if we're able to get new and better and more resistant prone drugs against gram-negatives, we could avoid 11 million deaths between now and 2050. So it's something that is a huge need. It's a big challenge, but it's something where we need to think really differently. And that's also one of the ideas behind the tripartite, is that we as three organizations should be able to come together and try and tackle the real huge issues. That is not something that is easy to see, success immediately, but something where it's really needed to do things differently. And that's what we're trying to do here. We want to bring together researchers from academia, from industry, And we want everybody to work together in a more collaborative fashion and in a consortium where there's open data sharing. So we don't have everybody sitting in their own silos going down and doing the same types of repeated library screenings or whatever, but to try and really come together and define some more innovative approaches to how we want to attack drug discovery, trying to hopefully use some of the more new enabling technologies like AI or different types of biological screening methods that we can use to be much better at identifying the relevant drug targets. And we really want people to try and tackle this, not just from trying to make incremental improvements like has been seen in the past on existing drug classes, but really try to come up with something that's entirely new and hopefully also be able to try and figure out and how can we actually develop something that would also be less prone to develop resistance quickly, because that's obviously the inborn problem here is that, as soon as if we were to develop a new drug and market it, you always run the risk that after a certain amount of time, then the bugs again will outsmart us and figure out a way how to become resistant to this new drug. So it's also about trying to really construct medicines in a way that will hopefully make them less prone to resistant.
Mm-hmm. Mm-hmm. And a lot of what you were just mentioning there, and we'll put the link to the RFP site on the grand challenges, so people can see a lot of these details. But what is, leveraging what you were just talking about, I found it quite interesting that in the sort of the list of the objectives, we talk obviously, looking at both chemical, AI, and biological tools, and then you mentioned in here that proposals can also address combinations or agents that hit multiple combinations. And I think this is, obviously, as you were talking about the Efflux pumps, when it comes to the way these organisms, evolve and find all these interesting ways around our antibiotics. Are you also looking, because, it's called an antibiotic challenge, but are you also considering things that may, for instance, just impact an Efflux inhibitor and work in coordination with an existing antibiotic to make it more efficient? Or also, I know a very hot topic recently for us has been sort of the quorum sensing and, the virulence factors, the stuff that, okay, it doesn't, it stops it from, coming together and climbing and sticking and all that, might not affect the bacteria itself. But these combinations are quite interesting to me and I'm just interested in how you think about, what a potential combination strategy could look like in this challenge.
Yeah. So I think first a disclaimer that I'm not a expert microbiologist in this particular area. And that's an important kind of viewpoint of mine, is also that we as funders, we can't be experts on all the things we do, but we can actually, make sure we engage with all of the experts out there, and we can make sure we scope out and hopefully get the best research to tackle this. So that's just a disclaimer I wanted to make first. But we have had a lot of discussions in the group about how to do this, because it's, at a certain point in time, you also want things to move ahead and you want to be able to do things. There's been this also discussion a lot I've seen on, okay, we want small molecule, direct acting small molecules, but what about all these other new and interesting types of modalities? Right. And I think it's, in general, that's something that we as a foundation and within the AMR community to try and solve for this. There are a lot of other types of modalities that are super interesting, including those where you combine things that actually work more as an antivirulence factor and not so much as a direct acting drug, etc. But I would say for the purpose of this initiative, it's been important, and that's also very much from an affordability and accessibility point of view that we want to also make sure that this is something that can also be actually scaled up and used at an affordable price across many settings, and those that are the hardest hit are obviously lower resource settings. So the small molecule direct acting target is also very much from a perspective of sort of affordability and ability to be able to produce it and deliver this at scale. And it is also to try and start to focus on something, because it's also, if you want to have a consortium together to really tackle a problem, I think it becomes very difficult if you've got people working on different types of peptide solutions, on bacteriophages and all of that. That's very difficult. And also, I think it's important, and it's something that is really a big issue here, is that the whole development pathway we have afterwards is relatively well established for, small direct acting small molecules, because assays have been validated. Like
Right.
that pathway is kind of known, but for a lot of these alternative modalities, a lot of extra work has to go into really defining how is that development and how are the assays and how there's all the things that happens around the actual, drug design and development itself that is not as mature yet. So, just to say that there's a lot of different considerations in terms of how do we define the actual scientific problem to tackle. And those also do consider how is things actually able to be applied down the line, how are things able to be developed down the line, etc. And that's one of the reasons why we have simply chosen this topic.
Mm-hmm. Mm-hmm. And it also mentions, along those lines that obviously the primary target is Klebsiella, but you of course you'd be interested in things that broadly are going to impact this Enterobacteriaceae family. Do you foresee? I mean, I assume we would know about them by now if they existed, but that maybe because the topic of repurposing has kind of been hot for us as well lately, and I don't know how many sort of preclinical to sort of early clinical development antibiotic candidates are sitting somewhere in pharmaceutical cabinets somewhere that just weren't developed because for one reason or another. Do you foresee sort of a repurposing component to this, or are we really starting at scratch, sort of drug development, drug discovery 101, and what do you think the balance might look like?
I think it's no secret that we also do hope to learn from those experiences that are sitting there. If nothing else, it'll be really nice to get those inputs to say what didn't work, because then we don't need to focus on that. And as you mentioned, it's really important that this new drug hopefully would be covering more broadly across the gram-negative. So the idea to focus initially primarily on Klebsiella is not to say that we just want a drug to treat Klebsiella. It's to make sure that the first round of activities that happen, the first consortium that is built where people try and tackle this problem from different angles, is focused on one organism, because then that can then set up the framework and then expand it. But it's also the hope, I would say, that whatever would work against Klebsiella, it's not unlikely that that would also be able to cover some of those other gram negatives that have similar challenges, or at least that that design process or discovery process could be transferred to also inform how can we do that better across the family of organisms. But I think it's the hope, and that's also why the consortium setup is the way it is, and the data sharing requirements is like it is, because we do hope that those previously learned lessons or libraries sitting somewhere that, we would like to learn from that. So we hope to bring people that have worked on this and have experiences to share into the consortium as well, for sure.
Mm-hmm, mm-hmm. And before, you brought up bacteriophage, which, obviously that's a tricky tool, but it's one that, I've covered a few times on the show, and it's just interesting to me, thinking about the organisms like Klebsiella that have evolved a long time to deal with things. Well, what are the organisms that have evolved even longer than them? The viruses. Bacteriophage might not be part of this program, but I think it's kind of cool. But what other tools? Again, coming with the caveat that okay, neither of us are infectious disease specialists, but what else excites you in terms of potential topics, whether it applies to this directly or maybe tangential to the whole area of anti-infectives, the big AMR issue? Is it bacteriophage? Is it again, so we talked the virulent factor interfering substances? Looking out to the horizon, what else gets you excited about possibilities in this space?
I actually we have, it's no secret that I think vaccine holds great promise also against AMR for obvious reasons, including, just preventing infections in the first place, both susceptible and resistant ones.
Right.
Also not being as prone to develop resistance, you could say. Vaccines are much more robust than antimicrobials are. But I think there is a gap because there are some of the really high threat pathogens within AMR that we don't seem to be able to really tackle with vaccines. So there's been a lot of effort trying to develop vaccines against Pseudomonas and Acinetobacter and things like that. And it's just a very tricky target, but it's also a very tricky population to target for.
Yeah.
So it's not as easy. For a big primary prevention vaccine trial, it's relatively easy to identify who is the population you want to vaccinate, where do you want to introduce it, how do you want to do it, and measure it. But for some of these trickier pathogens, which cause a huge amount of disease burden through hospital acquired infections or healthcare associated infections, it's not as easy to say this would be the target group, because you would have to predict who is at risk of going into the hospital and getting treatment and therefore getting, a resistant infection with this through a ventilator-associated pneumonia or whatever it is. So it's a much harder thing to attack. And one thing we have been discussing a lot, and we've also started talking to different people around the world, is this, the promise of monoclonal antibodies, which is sort of a thing in between, where it's not a primary prevention. It may be something, and it's something, I mean, this is not an easy thing to tackle either, because they're quite expensive to produce and it's difficult. But this is something where I think maybe there could be an opportunity here to get that, because we've seen sort of extended half-life monoclonals, and we've also seen progress in innovation in terms of making these much more affordably produced. And so I think there could be an opportunity here if we could develop some monoclonal antibodies against these really difficult to treat resistant bugs that cause a lot of the hospital associated or acquired infections. There could be sort of the in-between, where you would be able to give something that's similar to a vaccine would prevent these things, but it's not like you have to treat it afterwards and have all those challenges. So that's something that could sort of or bridge the gap, I think, a little bit between vaccines and the antibiotics that we have to give after people have gotten the infections.
Yeah. I appreciate you bringing that up. I don't always mention the monoclonals, but yeah, that's clearly fits a space there that is part of the armamentarium. So no, I appreciate that, and we don't talk as much about that as we should, but no, that's great. So I mean, the announcement was a month or so ago. I mean, the RFP opened February 11th. There's a deadline coming up now, March 25th. Could you tell us what happens next, and a little bit of whatever you could say about sort of how many groups have applied so far, and sort of what some of the next deadlines are going to be for 2025, and anything else coming up that we should know about per the challenge?
Yeah, I don't have the final numbers yet. As you know, the call hasn't closed. It will be closing on Tuesday next week. But what I can say is that the interest has been massive. So we have a huge number. As it looks right now, we're going to be receiving a large number of applications, which is really exciting, and we really look forward to diving in and seeing what great ideas have come up, and what research groups and who are the different people wanting to help tackle this challenge. So it's a bit too soon to say, but I can say that so far it looks like the interest has been very, very big. And that's very exciting. And then there will be a process of obviously looking through all of these. We've convened an expert panel to help with that again, to make sure we have the brightest minds and the best eyes to really make sure we can read and take it all in and figure out how to go from there. And then through the spring, there will then be the evaluation process and the decision on. So it's a two-stage process. So the first is an expression of interest, and then based on that, we select a number of applications, or full applications after that. But too soon to say exactly when that will be. But we definitely aim to get moving during the spring with this.
Awesome. Yeah, no, it's great to hear there's a lot of excitement and a lot of proposals going in, because clearly this is a major issue, not just obviously Klebsiella, but the entire AMR front. And again, it's exciting to see you leading the effort here and moving initiative of this scale forward. So I applaud you, and again, wishing you the best with the program. And I look forward, hopefully, you can do a follow-up a little bit down the line to see how everything is going.
That would be great. I think we have an obligation, because we see not only a sort of dwindling pipeline within the anti-infective space, but we also see a dwindling amount of people working on it, which of course is related. So we really hope to be able to keep this good work going and encourage and reinvigorate the work on anti-infectives. Because if we don't have effective antibiotics, there's a number of other disease areas, including cancer research, to come back to that, which, if we don't have effective antibiotics, cancer treatments are going to be much more challenging. So it is, antibiotics is basically something it's sort of what holds a lot of other medical treatment and care up. So we really need to make sure that we can keep innovating in this space and bringing more products to the market, but certainly also doing a number of other things within AMR to make sure we hopefully can curb this really worrying development that we're seeing.
Yeah, absolutely. Absolutely. Oh, really, really great story. And it really has been an honor listening to your journey, and I'm happy that you're heading this up. Again, for everybody that is going to be listening to this episode of the show across the various podcast networks or watching on the YouTube channel, again, you've been listening to Dr. Marianne Holm, Vice President, Head, Infectious Disease Programs, Novo Nordisk Foundation, really doing amazing things to push forward the gram-negative antibiotic discovery innovator. We'll put a link to both in the bio notes of the show. But Marianne, again, I want to thank you for taking the time out of your schedule to talk to us for a little while today about what you're up to. Obviously, thank you for what you're doing on the AMR front. And as we like to say on our show, thanks for helping to create a better and much safer tomorrow via what you're doing for all of us. Really a great story.
Well, thank you so much. It was a pleasure to be here.
Great having you.